Thank you. Yeah, I'm Meike. I'm, as you said, going to talk about a cortical coordinate system for psychiatric vulnerability. So throughout the past few years, we've gained quite some insights into shared pathological brain alteration that we can observe across different disorders, both at the regional level, like the insula, for example, but also at the level of more global alteration patterns that are spatially correlated between some groups of disorders. Now, these overlaps likely emerge from shared or rather unspecific risk factors and common neurodevelopmental alterations and contribute to the overlapping symptom profiles and comorbidities we observe in the population. Now, the question we've been tackling is how pathological brain alterations, which probably do not occur in isolation, but rather in a network-like fashion, are organized across the cortical landscape, and which features of the cortex leave some brain regions more vulnerable than others. So there are different features that may influence cortical vulnerability, like, for example, elevated levels of metabolic stress in connectome hubs, different levels of plasticity associated with cytoarchitecture and myelin, or the regional expression of certain risk genes. So overall, the cortex is a heterogeneous structure, and it's likely not just randomly attacked by pathological alterations. At the same time, cortical development is heterochronous and associated with varying plasticity levels. So unimodal sensory regions mature relatively early, but transmodal association areas show rather protracted development well into early adulthood, which also means they have a longer plastic period that facilitates, again, susceptibility to environmental influences, potentially adverse environmental influences. So today, I'll first talk about a cortical coordinate system, so kind of a spatial approach to capture cortical vulnerability, and then talk about a particularly vulnerable period for the emergence of psychiatric symptoms and focus on adolescent susceptibility to environmental risk factors and brain maturation. So for the first study, we used Cohen's D maps, so meta-analytical cortical thickness alteration maps from the Enigma Consortium for 6 psychiatric disorders. And to find systematic transdiagnostic co-alteration patterns, we computed a co-alteration matrix, which is basically the correlation between Cohen's D values between regions and across disorders. Or to put simply, we wanted to see if any 2 regions tend to be affected to similar degrees consistently across disorders. Based on this, we computed a co-alteration hub map, which reflects regions that are where cortical thickness alterations are particularly strongly coordinated with the rest of the cortex. And these co-alteration hub map, or these hubs themselves, were co-located with normative functional connectivity hubs, indicating a certain role of nodal stress. So here, the idea is that regions that are highly interconnected and highly engaged in many different subtasks tend to be more metabolically stressed and therefore more vulnerable to pathological alterations, being one potential mechanism shaping this pattern we observe. Then to expand on this connectome approach, we also wanted to see if there are some regions that are suspiciously strongly connected to these co-alteration hubs and may shape this cortical alteration pattern through their connectivity profiles as potential disease epicenters. So we used structural and functional connectivity data from the Human Connectome project and observed most prominent epicenters in ventrolateral prefrontal and medial temporal regions. These also overlap between functional and structural modalities to a certain degree and are generally found in regions that exhibit rather long-range connections as well as protracted development, both kind of facilitating a potential contribution to shaping a cortex-wide pattern. In a second chapter of this study, we wanted to identify cortical axes along which co-alteration profiles might be organized using a gradient approach. So these 2 axes, you can think of them as kind of opening up a spatial coordinate system across the cortical landscape. And the easiest way to read them in this context is that regions that are plotted with a similar color on the brain here tend to be affected together across disorders. So for example, looking at the first axis here, 2 prefrontal regions likely show similar co-alteration profiles, and a prefrontal and temporal region, not so much, or at least not consistently across disorders, and we are interested in a transdiagnostic coordinate system here. The second axis mainly differentiated unimodal sensory and limbic from heteromodal occipitoparietal regions. Now we, of course, wanted to understand what these axes actually reflect, so we first looked at the neurobiology, or what we know about the cytoarchitecture of the cortex, and stratified the 2 axes accordingly. And what we saw was that regions with maximally different illness effect covariance are regions with maximally different cytoarchitecture. So the first axis here segregated thick cortex with large cells from thin cortex with many but small cells. So this suggests that our gradients or axes here might, on the one hand, differentiate or different plasticity levels associated with maximally different cytoarchitectonic profiles. And we also know that regions that show a more similar cytoarchitectonic profiles tend to be more structurally integrated and connected to each other, potentially giving us a structural scaffold for the co-alteration network we observe. In a similar approach, using the Neuroscience database, we wanted to see where functional networks are located along these two axes or in this coordinate system. And we observed that the levels of processing a region is involved in determines which regions tend to be affected together as a transdiagnostic feature. So we see differences in illness effect covariance in regions processing or involved in basic sensory compared to attention perception, compared to more active cognitive control tasks and more abstract cognitive control tasks. So it seems like brain regions processing similar types of information, for example, sensory information, seem to show a similar vulnerability level no matter where they are located on the cortex. So overall, by investigating the embedding of cortical vulnerability in a transdiagnostic context, showed us that cortical alterations seem to be coordinated across the cortex and across diagnoses along connectomic, cytoarchitectonic, and functional dimensions. Now, while the first study took a cross-sectional and kind of spatial approach, these patterns, of course, do not appear out of nowhere, but we assume that there's a developmental road leading up to these patterns. So as I mentioned in the beginning, the multi-modal organization of the cortex is not only associated with potentially different vulnerability levels, but also with different maturational trajectories. So for another project, we specifically focused on a vulnerable period for the emergence of psychiatric symptoms and tackled the question how delayed consolidation of association areas during adolescence links to intra-individual trajectories and susceptibility to environmental risk factors. So we basically sampled adolescents or young adults' responses to different loads of environmental stressor exposure at 2 time points, roughly 1 to 2 years apart, and then linked potential behavioral changes to changes in ongoing brain maturation. And here we focus on a central feature of adolescent brain maturation, which is myelination, which I like a lot because it's both plastic in itself. It generally increases during adolescence and it kind of adjusts network dynamics on the go. But it also, of course, functions to reduce plasticity by consolidating connections once they're established. So this double agent role of influencing both plasticity and stability in a way makes it super interesting to look at in the context of behavioral adaptation. For this project, we used longitudinal data from the neuroscience and psychiatry network cohort. Here, individuals were between 14 and 26 years old. And as a first step, we derived a general distress score as a latent factor derived from several transdiagnostic mental health assessments. Could also be referred to as a P factor. We then, in a separate subsample to avoid leakage effects, predicted this general distress score from adversity measures, including adverse life events like death of a family member, for example, childhood trauma, difficult family situations, and socioeconomic status. Now, conceptualizing more resilient or more susceptible outcomes as better or worse than expected well-being levels given the adversity someone faced, we extracted the residuals from this model as our resilience scores. So maybe an easier way to think of this is that it's a continuous score predicted or derived for each time point of each individual, which kind of reflects mental health well-being adjusted for inter-individual differences and also intra-individual differences in stressor exposure loads. Now, linking potential behavioral changes in the stressor exposure response to ongoing brain maturation, we first observed a higher rate of prefrontal myelin-sensitive MT change in the anterolateral prefrontal cortex in adolescents that developed towards more resilient outcomes, whereas we saw an attenuated or like a lower rate of myelin increase in individuals that became more susceptible with age. We then wanted to see if this enhanced consolidation that we would assume with increased myelination also coincides with effects on prefrontal functional network embedding. So we performed a seed-based connectivity analysis and observed generally more maintained levels of prefrontal network embedding, primarily within the default mode and frontoparietal networks, in individuals that became more resilient with age, whereas we saw a segregation in these networks in individuals that became more susceptible. So note that there's this co-occurrence of more functional change and less myelin increase in individuals who become more susceptible. And lastly, we wanted to acknowledge that individual brain regions usually do not mature in isolation, but in a globally synchronized fashion. So we also took a system levels approach here. And for that, we used regional profiles of our myelin marker. So we sampled the empty intensity contrast, which approximates myelin, across 10 intracortical depths, which gave us regional microstructural profiles. Based on this, we could compute a microstructural profile covariance matrix, which basically captures the interregional similarities of these profiles. And based on this again, we can assess how individual embeddings of certain regions change with age, and if this change is a reorganization of existing patterns or a strengthening of existing patterns with age. The idea behind this microstructural profile covariance approach here is that regions that tend to become more similar to each other in their microstructural profiles with age tend to become more structurally connected to each other. Now, this method mainly captures two maturational modes, here plotted for the full sample. And here in blue, we see disruptive reorganization in a frontoparietal heteromodal midline, which means regions that were more similar to each other tend to become more differentiated with age, or vice versa. So there's a lot of reorganization happening here, whereas this is framed by ventral and dorsal conservative strengthening of existing patterns here in red. Now, we of course wanted to see how these modes differ in individuals that showed positive versus negative changes in their abilities to cope with stressors. And we generally observed a bit of a negative shift towards more disruptive reorganization and less strengthening of existing patterns in individuals that became more resilient with age. And these effects were not systematically located in regions that generally change most during adolescence, looking at microstructure, but rather in regions generally located higher up the cortical hierarchy towards heteromodal and default mode regions. To sum up, we observed a role of cortical refinement primarily in association cortices that's not only the more the better, but macro and mesoscale cortical refinement happening in parallel. So looking at microstructure, we see a higher rate of prefrontal myelination and more global reorganization in individuals who became more resilient with age, which was accompanied by more stable functional networks. On the flip side, we saw more functional instability and kind of less myelin maturation in individuals that became more susceptible with age. Together, what we learned from these two projects was on the one hand, that the structural and functional similarity between regions, like their cytoarchitectonic profiles, the types of information they process, determines if two regions show a similar vulnerability level, and this is independent from where they are located on the cortex, which kind of gives us this transdiagnostic vulnerability landscape. And at the same time, the timing of plastic periods during development varies across the cortex, and the consolidation of particularly association areas here influences individual trajectories of susceptibility to environmental risk factors. So it seems like it's both the spatial arrangement as well as maturational trajectories that influence cortical vulnerability and give us this cortical coordinate system, or what we refer to here as a neural p-factor. With that, I'd like to wrap up. Thank you all for your attention, and special thanks to my supervisors, Sophie Falk and Simon Eickhoff. Thank you.
Excellent. Thank you so much. We have time for one question, please. Anybody?
Yep. Thank you for the amazing talk. I was wondering, since many of the results you showed are on the group level, how do you make this jump from the group level and the meta-analysis level that you take from the Enigma studies to the individual level into vulnerability?
Mm-hmm. You're talking about the first project? Yeah. Actually, we're doing that at the moment. Good that you ask. So what you can also do with these axes is embed individual disorder maps within this coordinate system, but also, of course, individual level maps. So, for example, we see that individuals with autism spectrum disorders, their cortical alteration profiles align very well with the first axis, whereas the second axis captures, schizophrenia, bipolar disorders a bit better. So you can also kind of take the cortical thickness map of an individual and embed it in this coordinate system, which then again gives us kind of a continuous transdiagnostic space where individuals are located in this space. Thank you.